GenAmplify Corona Virus Disease-2019 rRT-PCR Detection Kit Tutorial Series.
Extraction of COVID-19 viral RNA from respiratory sample. Consumables and equipment needed. Reagents and supplies. RNA extraction kit. Disposable powder-free gloves. P20 or P10, P200 and P1000 micropipetors and aerosol barrier tips. Sterile, nucleus-free 1.5 ml microcentrifuge tubes. Laboratory marking pen. Racks for 1.5 ml microcentrifuge tubes. Absolute ethanol 96 to 100%. 10% bleach and 70% alcohol for decontamination. Paper towels. Appropriate waste container for used tips and tubes. Before extraction, follow proper donning of PPEs. Decontaminate the BSC area following this protocol. Decontaminate surface with 10% bleach from the center going outwards. Decontaminate pipetors with 10% bleach from the tip to handle in a single direction. Decontaminate tip boxes with 10% bleach from the mouth going backwards. Decontaminate surface with 70% ethanol from the center going outwards. Decontaminate pipetors with 70% ethanol from tip to handle in a single direction. Decontaminate tip boxes with 70% ethanol from the mouth going backwards. Change gloves before preparing reagents to be used. Prepare the consumables and reagents from the GenAmplify Viral RNA Purification Kit. For kits opened and used for the first time, perform the following steps. Buffer preparation. Add absolute ethanol to buffers RB1, RBW, and RNW. Check individual buffer bottles for the correct volume of absolute ethanol to be added. Mix gently by swirling. Properly label the bottles with the name or initials of the person preparing the buffers and the date when the buffers are prepared. Carrier RNA preparation. Add 370 microliters of nucleus-free water to the tube containing lyophilized carrier RNA and dissolve thoroughly by flicking. Transfer 7 microliter alicos to clean 1.5 ml tubes and store in the -20° freezer until use. Proceed to extraction of SARS-CoV-2 viral RNA from the respiratory sample. To the thawed 7 microliter alico of carrier RNA, add 300 microliters of buffer NVL. Properly label the tubes according to sample ID. If precipitate is formed in buffer NVL, heat it to 56 degrees Celsius to dissolve before use. Sample lysis. Transfer 200 microliters of each sample into a labeled carrier RNA NVL tube. Mix thoroughly by pipetting in and out 10 times, followed by tapping the tube with your forefinger 10 times. For proper lysis, the complete mix of sample and buffer NVL is essential. Incubate the mixture for 10 minutes at room temperature. Add 350 microliters of buffer RB1 to the mixture and mix thoroughly by pipetting in and out 10 times, followed by tapping the tube with a forefinger 10 times. Unpack appropriate number of spin column and properly label them according to sample ID. Carefully transfer up to 750 microliters of sample buffer mixture to the labeled spin column. Close the cap and centrifuge at 10,000 xg for 30 seconds at room temperature. Ensure that the solution has completely passed through the membrane. If not, centrifuge again at a higher speed or at longer duration until all solution has passed through. Discard the pass-through and reinsert the spin column back into the same collection tube. If the mixture volume exceeds 750 microliters, repeat the previous step with the remaining solution. Washing step. Add 500 microliters of buffer RBW to the spin column. Close the cap and centrifuge at 10,000 xg for 30 seconds at room temperature. Discard the pass-through and reinsert the spin column back into the same collection tube. Add 500 microliters of buffer RNW to the spin column. Close the cap and centrifuge at 10,000 xg for 30 seconds at room temperature. Centrifuge at full speed for an additional 1 minute at room temperature to remove residual buffer. Care must be taken at this step for eliminating the carryover of buffer RNW that can interfere with the downstream reactions. If carryover of buffer RNW still occurs, centrifuge again for 1 minute at full speed with empty collection tube. Elution step. Prepare new 1.5 ml tubes with cap and label according to sample ID. Transfer the spin column to the labeled 1.5 ml tubes. Carefully add 30 microliters of nucleus-free water to the center of the spin column. Let it stand for 1 minute at room temperature. Close the spin column cap and centrifuge at 10,000 xg for 2 minutes at room temperature. Ensure that the tube containing eluted viral RNA is tightly capped. Post-extraction procedures. Follow proper doffing protocol for PPE. The most contaminated PPE will be the secondary gloves. The face mask and primary gloves will be the PPE that will protect the personnel up to the last stages of doffing. Store extracted viral RNA in the fridge. Dispose all materials following proper disposal protocol for infectious waste. Decontaminate the surface and all equipment after use by wiping the surface with 10% bleach and 70% alcohol. Fully close the sash of the biosafety cabinet and turn on the UV for at least 30 minutes. Use a disinfectant spray to disinfect the working area before leaving the sample processing area. Proceed immediately to the next step, which is the COVID-19 rRT-PCR assay.
Brought to you by Manila Health Tech Incorporated, Technology Simplified.